This is my third take, even though I said that I wouldn't do 3 takes. I think this is going to be my final one if I mess up. This is me doing what I've learned from the textbook. Watch at your own risk because I'm assuming you've already looked over the slides. This is my disclaimer. You can pause and read it. I don't care. These are the 4 points I'm going over. You can read. Here's the absorption of amino acids. I realize that I've done this wrong, fortunately, but I'm just gonna go over the points, and I'm sure you can also figure it out yourself as I go on. So this is how amino acids move, as well as the ions. So it starts in the intestine. I'm pretty sure it's the small intestine as well. Yeah, the small intestine. These are going to be really bad because I'm using the PowerPoint pen, and it's not really good. Um, so there are 6 sodium dependent transporters, and you're probably asking why are there 6? There's 6 of them because the amino acids that they push through, there's multiple of them, but the amino acid that they push through, it has a preference. So it could take one of them could take one amino acid, but it could take 2 of them, and then I don't know, but it could prefer one of them, and then that's why the second one is there, so that I can push that other one through, even though it might have a preference for another one. It's just redundancy to push stuff through, but he probably won't ask that, just for your knowledge. Um, this is the part that is wrong that I realized. So I wrote that the primary transporter depends on the sodium gradient, and the secondary transporter couples amino acids with sodium. I originally thought that this one right here, oh god, that this one right here is the primary one and this was the secondary one. I'm inclined to believe that there's only one of them, and this is the secondary one. This is the one that couples it because obviously there's an amino, you can see that there's an amino acid going with the sodium ion into this thing. I don't even know what this thing is. I'm gonna be honest because I'm just trying to explain this picture that was put on the slide. Um, so the amino acids go to the serosal side, um, and serosal blah 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 blah. The serosal membrane. This is the serosal membrane right here. It's a thin 2-layered tissue that lines the intestine. So it's going from the lumen to, like, I'm assuming the edge of the intestine. I don't know. I told you, don't ask me questions. The amino acid go to the serosal side into interstitial, sorry, interstitial fluid. So the facilitated transporter right here is bidirectional. And the reason it's bidirectional is a little morbid because if you're pushing amino acids through, you need the amino acids obviously to be broken down eventually, because that's what part of this lecture is for, I think. But if you need to bring back the amino acids or something, if you need to bring it back to going this way because your cell is starving itself, it can go back up that way. Um, that's what I understand. I don't know. It just says it takes the amino acids from the blood if it's starving. So, moving on, the active transporter is called I'm sure you can figure this out, sodium potassium ATPase, and it pushes sodium out into the basolateral membrane. This area is the basolateral membrane because we can tell that it's going outwards. That's where the sodium's going out and the potassium's going in. So it ends in the blood. This is the blood because you can see it says vein. Where else has blood? In your veins. I mean, there's other places, but like the vein has what? Blood. And then it eventually goes to the liver. Um, and this whole picture describes a trans epiphysis. acid transport. Trans, cross, epithelial, skin, amino acid transport. That's how it's moving. Um, oh god, it stays. Okay, great. The pool party. Here's the pool party. So what is the pool? So the intracellular amino acid pool is generated from, oh god, diet proteins. This really doesn't like my pen. I'm so sorry. It says diet proteins. And protein degradation. I thought this was going to be better. Proteins are always being degraded and damaged by oxidation. You can see there's a pool there that I've added. I don't know. I'm sorry. So the lysosomal and ubiquitin protease, oh god, yeah, protease system is meant for the degradation or of damaged or unnecessary protein. So what is this pool for? This pool is for the total concentration of free proteins, I'm pretty sure. Yeah, no, no, of free amino acids. I didn't study before I did this. Sorry, guys. Um, Free meaning that they can be used for, oops, synthesis, can be used for energy, and they can be used for oh my god, metabolic regulation. I really hope you can read that. I'm so sorry. You can um and then what amino acid is there? I just thought it was fun to put in. That is glutamine, I think. I can't even remember. But the reason I put glutamine there is because it's important to this whole lecture. Now you're probably thinking, James, I don't know anything about those things. I know, neither do I. This is what we know so far about lysosomal um, protein degradation and the ubiquitin proteasome pathway. Nothing, probably. You should probably know some, though. So let's start with lysosomal, because that's the much easier thing. So lysosomes participate in autophagy, phagy, I don't know. And we can figure out what that means because auto is self, and that other word is just eating. I too eat. The process is regulated, so it doesn't just go on all the time. And the process begins when the cell starves itself. I know, it's like, why is everything starving itself? So the lysosomal thing is also ATP independent. Psych. You thought it was dependent. Um, there's no ATP involved, from what I understand. And the important family of proteases involved is called. You sound it out. Ethepsin. That's what I'm calling it. And a protease, for a reminder, a protease is an enzyme that breaks down proteins and or peptides. That's it. That's basically it. So you're probably like, whoa, what's this? I've put a square there because it's for lysosomal. You can fill in the blanks as well. So cytoplasm, so it starts with cytoplasm being sequestered. I don't know what sequestered means. I had to look it up. It means isolated. Cytoplasm's isolated into vesicles. I don't know what that means. That's just what I wrote. Into vesicles, and then delivered The where? The lysosomes, because that's what we're talking about, the lysosomes. Oh my god, that's an E. Now we're in the lysosome. So then something degrades the protein into its individual amino acid. Trusty friend cathepsin to the rescue. Remember, cathepsin is the protease that's involved in breaking down the protein. This is 2.5-ish. This is how we're gonna go. So the amino acid then leaves the lysosome. Where does it join? The pool. That's where we want it to go. The pool, technically the amino acid pool, if we really want to be more specific. It's right there. Cellular amino acid pool. And important to note, because these are the two things that he wrote down, pepsin, where does it work best? Acidic, basic, acidic. I don't know why. So this process, does it involve ATP? No, so it's ATP independent. So this is what we know now. Remember, lysosomes participate in autophagy, they eat themselves. The process is regulated. The process begins when the cell starves itself. It's ATP independent, and the important family of proteases that is involved in lysosomal like degradation or whatever is, it's on the screen, cathepsin. Okay, now we're going over ubiquitin proteasome pathway. I think this one is a little, I don't know, it's a little more confusing to me because there's not as much that I have written down for it. So ubiquitin is a small protein. I don't know why I wrote that down. It is highly conserved. I don't know what that means either. But that's what he wrote down in his slide. So just write it down again so you remember. And it covalently bonds to the what group? What is that thing? That is epsilon. The epsilon group of what? Of lysine. So what does that mean really? The ubiquitin proteasome pathway starts with basically like a gun, but you're not going to shoot that gun right away. You want to figure out who's your target. So in the protein, to the protein that you want to find, if this is our little protein, that's a protein. What does he look like? Okay, there's our protein. Okay, there's our protein. And you want to put a target. The ubiquitin part of this pathway is basically sorry, I keep looking at the picture. The ubiquitin part of the pathway is basically to start it. So it's starting, it's kind of getting the protein ready, and then the proteasome pathway is when the action actually begins. So it puts the target. How do you draw a target? Yeah, that's a target. A target on, um, just looks like he's blushing, but it puts a target on the protein that it wants to break down. So a damaged or a degraded or a damaged protein. It's a target on there with ubiquitin. That says ubiquitin, please. So this is your target is basically ubiquitin. That's why I put a little target there. So again, the protease is an enzyme that's used to break down proteins or peptides. So again, step one, the target. Step two is a complex. What complex? It's right there. So this is the protease. Oops. The protease complex. Sorry. So the protease complex can now degrade the protein because it's been targeted and it gets degraded into small peptides. And because it's been broken down by these small peptides, it kind of like frees up that target. Like, think of it kind of like a physical target that you've put on, and now that it's broken down, you can pull it back and use it again. So, what was our target? Ubiquitin. So, it releases ubiquitin again, so it can be recycled. Go. And another important to note, remember, ubiquitin proteasome pathway is a circle. I've used circles. And the other one is a square. I've used squares or rectangles. Don't argue with me. So in order for the protein to unfold, ATP hydrolysis is used, which means that the process is ATP dependent. And now your protein that has broken down from small peptides can join the pool. So now this is another slide. Which protein goes where? Can you tell me those letters? Probably. This is like an eye exam. So the proteins that are rich in proline, glutamate, serine, and threonine follow which pathway? I don't know. Yeah, I do. Just kidding. They follow the ubiquitin protease pathway. Okay, we're gonna do this again. Ready? Do it with me. Okay, ready? The proteins that are rich in proline, glutamine, serine, threonine follow the UP pathway, the ubiquitin protease pathway. I'm not repeating, I'm not writing that out. You already see how much I'm struggling to write. Okay, amino acid catabolism. This is the longest part. What's the purpose of amino acids being in the pool? Go back, figure it out, come back. Okay, it is to synthesize proteins. That's why it's in the pool, right? Because we need those to function, right? Okay, good. Psych, it's actually there for other things too. So it's there for the carbon skeleton and nitrogen as well. So the carbon skeleton oxidized for energy. And the, oh my god, I've messed it up. Okay, just pretend that I've written oxidized already. So they can be glucogenic or ketogenic, and you should already know this if you've watched the lecture. The two ketogenic amino acids are lysine and leucine. That's kind of fun that they're both L's. There we go. And the other thing about nitrogen, what is that converted to? You remember? I don't. I had to look over at my notes. It's converted into urea. Yay. So, back to amino acid catabolism. You're probably gonna ask me, can you explain why he put this there? No, I don't know why he put that there. I'm just trying to read my notes right now. I feel like him. We know that it starts with the dietary proteins, and then remember that like funny thing that looked kind of like this? Ooh, and there's a circle there. Ooh. And that way this sodium went out, the potassium went in. Oops. That is what's going on right here. This part, I don't really know. This seems like I don't really. I've put it in so I could tell you that I don't really know much. Um, I'm just trying to relate this to what we did earlier. That's as best as I can tell you. Sorry. Um, okay. I think most of that is in the slide anyway. So, nitrogen removal from amino acids. So, the removal of the alpha amino group is called, that's not how you spell it. Sorry. Uh, transamination. There we go. Transamination. Oh my gosh, what did I just do on my tablet screen? So that's removing the alpha amino group. That's essential for the catabolism. That's what we're just talking about. That's what this said. Catabolism. Oh, wait. No, this is the subgroup. My bad. The catabolism of amino acids. Go do that part again. Go fill in those blanks. Okay. This right here. Actually, I'll wait for you. Just pause it. Go back and do it. Okay, we're back. So this picture, I know we put it on the slides, but it's pretty vague what it's talking about, and it took me a while to understand it. Maybe it didn't take you long to understand it. I don't know. I'm me. Like, piss off. This is the basic form of transamination. Right. You're starting with an amino acid, and then like I don't really know what this one thing is. So this is my diagram. Instead of that, this is my diagram to you. We are so back diagrams, and now we're doing triangles. Ooh. So your first amino acid loses its nitrogen. Do I know if it's an amine? No, I don't really know. I'm just going to call it a nitrogen group because that's what I know that it is. So the nitrogen group gets picked up by what? By the alpha keto acid. That's what I understand. Um, this is not the actual name of it, but this is the generic name. And then the N group change sorry, this is wrong. The alpha keto acid changes into its respective amino acid. What does any of this mean? Okay, let's see it. Okay. This was explained to me, broken down to me by my lovely girlfriend Joanna. Hi, if you're watching, you probably are. So oh, let's start with what I just said. So we lose our nitrogen group, and oh, this is what I was going to try and do. Okay, so basically what I understand is you have your uh you have like your amino group. Let's see. I think it's like this. Yep. Plus your, it's like put on, it's like your amino group attached to whatever amino acid. And then what happens is you add in your keto acid. And then what you get is your second amino acid that is different from this, plus your waste. That's what I understand. Okay, this looks really stupid. It will make a little more sense in the next one, I hope. This is the relevant one that he actually put in the lecture. Okay, let's see what I was talking about. So we're talking about this, right? We're talking this amino, this nitrogen amino group, right? Plus the alpha keto acid, and then a waste. And actually, let me just put this in brackets. It's, or quote, sorry. It's not actually waste, I think, but it is kind of like the waste, like the leftovers, right? So I'm going to call it waste. And then your second amino acid, I think. I don't really know. So there's our end group. And this is kind of the way that I understood it because these arrows didn't really make sense. So we have our aspartate plus the alpha keto. What is it here? Alpha keto glutarate. And then we want to take away the nitrogen thing. So then it turns, and then we want to give the nitrogen group to the alpha keto acid. So then what happens is this becomes, oops, oxaloacetate. Sorry. And then this becomes glutamate. And the thing that drives all of this is called PLP. You're probably like, what is that? That is sorry, I've written that wrong. Pyridoxal phosphate. So it's PLP, which is what makes it do its thing. PLP. So it's pyridoxal phosphate. I realized I just spelled it wrong down there. Ignore that. Okay, generation of ammonia. I don't really have much to say about this. The generation of ammonia occurs in the mitochondria, and the process is reversible. Okay, transport to the liver. What else do I have about this? Yep. Okay, so kind of the same thing because it's on the slides, but the most abundant amino acid is glutamine in blood. And glutamine is derived from glutamate. Glutamate's pretty important, but I don't know if he talks about how important glutamate is. I don't know. So why does this need to be transported to the liver? Why does the ammonia need to be transported to the liver? I don't know. I'm joking. So the urea cycle enzymes, the ones that break down the ammonia, are primarily active in the liver. And remember, we start where? We start in the intestine, small intestine. And then we're going through the blood, and then we're going to the liver, right? Okay. So if the enzymes are primarily active in the liver, what does that mean? It means the intestines express a high or low level of activity of those enzymes. That's why we need to move it out, right? Because they're not doing anything there. They're not doing anything significant enough. So, a side note, I know that his slide, one of the pictures, has alanine. The only thing that I read about it is that it's not totally relevant, but if you really want to know, alanine is just another carrier of nitrogen in the way that glutamine carries nitrogen. That's why it's the most abundant amino acid. I don't know. But that's the one that's relevant. And alanine is exported by muscle to the liver as opposed to glutamine using the blood to go to the liver. So this is the urea cycle. This is the base. This is the base. So this is the way the urea cycle is the way that amino acids are disposed, and 90% of the amino acids are disposed via the urine. Urea is produced in the liver. That's why we're going to the liver eventually. That's where hepatocytes are. I think that's where it happens. The urea cycle. Hep, hep, anything hep is liver. So the first two reactions occur in the mitochondria. I'm not writing that all out. And the last three reactions occur in the cytoplasm. So in total, if we do 2 plus 3, because that's all we do in this class is nothing, is 5. So there's 5 key enzymes. Okay, ready? Pause and go do it. You can do it. Okay, I'm assuming that you've done it now. So now the key enzymes. I'm gonna hold your hand through this, okay? The first one is carbamoyl phosphate synthetase. He wrote it wrong on the slide. The second one is ornithine transcarbamylase. That's the second one. He wrote the third one wrong and the fourth one wrong. Arginosuccinate synthetase, and then arginosuccinate lyase, and then arginase. You're probably not going to remember the full names, and neither am I. Okay, ready? Keep your eye open. I'm going to change the slide. Okay, much simpler. Start with CPS, OTC, ASS, ASL, ARG. Okay? Keep your eye open again. See, it's not that scary. It's the same thing, it's just shortened. We already know that, okay? Now, this is the stupid mnemonic that I've created. If your parents beat your ass, you're gonna call CPS, you're gonna get an OTC cream, an over the counter cream to apply to your ASS, and then the last two you're on your own, okay? ASL and ARG. Arg, like, it hurt to get my ass beat. I don't know. Whatever helps you remember the last two. I don't care. Okay? CPS, OTC, ASL, CPS, OTC, ASS, ASL, ARG. CPS, OTC, ASS, ASL, ARG. Okay, so I don't really want to go into depth about the urea cycle. This is as in depth as we're going to go. This is a drawing that I did in MS Paint, and we're just going to draw over this, okay? We start with NH4 plus, I think it's plus, I don't know. And that one thing from before, where is it? Aspartate. Oh, I've accidentally gone, oops, okay. Sorry. Let's go back to that, okay? Um, Going back to glutamine, let's see. Give me 1 second. Okay, so glutamine is the main carrier of nitrogen. We already know that because it's in his slides. And the way that glutamine gets there, um, I'm getting really tired, guys. I'm sorry. Um, Glutamine's derived from glutamate, right? We said that before. Somewhere. Yep. Okay. So we go from glutamate and whoop, we end up with glutamate and ammonia. Okay. This is the, yeah, I don't know. I'm sorry. The enzyme that goes from glutamine to glutamate though is called, can you guess? Glutaminate. I know it takes a pit stop in the liver and the intestines and the kidney. I'm gonna be honest, I can't remember why. The glutamine, I know that glutamine is fuel in the intestines. Um, and then this glutamate, oh, sorry, no, the ammonia helps with the urea production in the liver. I don't know. Sorry, I stopped us for that. Let's go back. Urea cycle. Um, so, like we said, start with following CPS and then get an over-the-counter cream or in the two steps because, like, it's not totally relevant. Get it for your ASS. And I don't know. Do you know ASL? I don't. Not really. And then you get your ARG right there. So CPS, OTC, ASL, ASS, cell, ARG 1, and then you get urea. Um, this is basically that really big. I don't know if he put a picture, but I've just slimmed it down, right? This is how I understand it. You start with CPS, and then to get to citrulline, you need OTC, and then through these two other steps that kind of it takes off the nitrogen to make urea, you get to arginine, and arginine gets broken down into urea via arginase. The arginine also helps regenerate OTC. I think. No, I'm not even going to say anything because I'm just going to confuse you more. I don't know what this meant. Like, why would you say fate of urea? Um, we're getting to the end, I promise. So, urea in the liver, uh, when you break it down, it can go through the blood or the intestines. So, when it goes through the blood, it's excreted by the kidney because we know the kidney does all the filtration. And then when it goes through the intestines, there's cleavage done to it via urease, what's called. CO2 gets released, and this is in the slides. Like the ammonia gets excreted in stool and also reabsorbed in blood. That's all we need to know. So the disorder of urea. Again, do I really need to talk about this? Like he already talked about it in the thing. It's in the name. Something that I read in the thing is that the brain swells due to NH3 toxicity. I'm pretty sure that's ammonia. I mean, come on. Hyper anemia. We all know what that means. We can all put together what that means. Um, when there's more NH3 astrocytes, what's it called? Anatomy 1 jump scare. It leads to more glutamine. And glutamine. What's that for? The carrier of, come on, please. The carrier of the nitrogen. Now it's making sense. Uh-huh. So basically to treat it, you just gotta reduce the amount of urease that's like leaving it because it's breaking down, right, into ammonia, breaking the ammonia down. So stop breaking it down, right? Or low protein diet or therapy like gene therapy or arginine therapy. So I guess just like lowering this so it doesn't convert into urea. That's probably what it is. And now we're done. Okay. Bye. I hope this helped.